The three related key topics are device-associated infections, biomaterials, and biofilms. They will be discussed in this sequence, followed by some plausible solutions. 1.1 The clinical problem, device-associated infection, DI is classified under healthcare-associated infection, HCAI, i.e. infection attributed to the interventions received from healthcare providers. Apart from creating huge economic burden on the healthcare system, these infections troubled patients with many complications, ranging from minor discomfort, longer hospital stay to serious disability and sometimes death. Second National Point Prevalence Survey (PPS) in Scotland reported prevalence of HCAI for acute hospitals and non-acute hospitals of 4.9% and 2.5% respectively. 22.6% and 39.0% of these patients in acute hospitals and non-acute hospitals had UTI infection, which made it the most common HCAI. About half of them were catheterized 7 days before they were infected. 10.8% of all HCAI was laboratory-confirmed bloodstream infection (BSI). 26.2% of these cases were secondary to peripheral venous catheter (PVC) or central venous catheter (CVC) use. Prevalence of pneumonia was 17.5% and approximately 1 in every 5 patients with pneumonia was intubated in the 48 hours before onset. Endotracheal tubes allowed pathogens from contaminated oropharyngeal space to migrate to bronchial mucosa, increasing the risk of contracting ventilator-associated pneumonia (VAP) by 6 to 20-fold. CVCs used for dialysis access resulted in exit site infection, bacteremias, tunnel infection or any combinations of them. The prevalence of PVC, urinary catheter and CVC use and intubation in acute care were approximately 33%, 20%, 4% and 1.3% respectively. Even if the percentages are small, given the large population of patients, they are translated into many patients. Patients with invasive devices have higher risk of developing DI. This is supported by a prospective study carried out in 15 skilled nursing facilities in southeast Michigan reported that residents who had urinary catheters and/or feeding tube had more infections than residents who did not have any of these devices. 331 infections per 1,000 resident months of follow-up versus 171 infections per 1,000 resident months. Reported relative risk was 1.3, adjusted for comorbidity, age and functional status, 95% confidence interval 1.1 to 1.5, p equals 0.002. Even though primary infection of orthopedic implants is uncommon, the complications are severe for patients. Since the pathogens in biofilms are not susceptible to systemic antibiotics, the only management is revision or secondary surgeries. The infected implant is replaced with a new larger one because bones are damaged when implant is initially inserted and subsequently removed. Surprisingly, reinfection rate following revision surgeries is higher than primary surgeries. This can be illustrated by an example of total joint replacement (TJR). Infection rates following primary and secondary TJR are 0.5 to 3% and nearly 10% respectively. This implies that revision surgeries resolve symptoms but may not eradicate the underlying pathogens. Despite aggressive measures, approximately 10% of unsuccessful cases lead to arthroplasty resection, arthrodesis or even amputation, which increase morbidity significantly. 1.2 Biomaterial in medical devices. Figure 1 showed 5 basic classes of biomaterials with some examples. Biomaterial is defined as non-viable material utilized in a medical device and interacts with biological systems intentionally. Biomaterials are usually integrated into implants or devices, instead of being used in isolation. They cover a wide range of materials and can be grouped into classes as shown in Figure 1. However, this list is not exhaustive. Different types of material confer medical devices with varying capability to resist bacterial attachment. Polyvinyl chloride or polyethylene resists bacterial attachment poorer than polytetrafluoroethylene, silicone or polyurethane. Silicone is the chosen biomaterial for our study because it has many medical applications as shown in Figure 2. This advocates that our work can potentially benefit many patients. Silicone possesses many superior properties ideal for its uses, i.e. non-toxic, flexible, stable chemically and thermally, biocompatible, biodurable, non-allergenic and permeable to gas. Hydrophobic silicone prevents blood coagulation, and its low critical surface tension of wetting, 24 millinewtons per meter, provide good surface coverage, making it ideal for anticoagulant coatings on needles, syringes and blood collection vials. Similar principles can be applied to coating other materials with silicone to give desired properties of silicone to other materials. Figure 2. Medical application of silicones. Silicone is fundamentally a synthetic polymer made up of many silicon-oxygen bonds as its backbone. Two R groups are bonded to each silicon atom. The basic repeating unit is siloxane. When methyl groups are the R groups, the polymer is called PDMS, which is the commonest silicone found. Refer Figure 3. Rotation energy barrier around Me2CO bond is extremely low, only 3.3 kJ per mole versus 13.8 kJ per mole for H2CCH2 bond, allowing PDMS chains to adopt many configurations. Generally, the chains orientate to expose the maximum number of methyl groups outwards. Weak intermolecular interactions between PDMS chains explains their very low glass transition temperature, i.e., 146 degree Kelvin versus 200 degree Kelvin for polyisobutylene hydrocarbon polymer analog. These factors confer PDMS high flexibility to follow the shape of body cavity. The invasive nature of medical devices creates a portal for entry for pathogens, bypassing the host's natural defenses. Pathogenesis involving different devices differ slightly as detailed in the next 2 paragraphs. 4 proposed pathogenic pathways for catheter-associated BSIs in the order of decreasing frequency are 1. Skin flora migration from cutaneous catheter tract and tip, 2. Intraluminal colonization following hub contamination, 3. Hematogenous seeding from other infection sources to catheters, and 4. Very infrequently, intraluminal contamination by infected infusate. 3. 9. For catheter-associated urinary tract infection, see a UTI, the first 2 pathways play similar roles. Since the catheters are not in contact with blood, the third pathway is not applicable. For the fourth pathway, instead of infusate, reflux of infected urine from the collection bag increases the risk of CAUTI. Bacteriuria greater than 105 CFU per milliliter is reached within 24 to 48 hours post-catheterization. In addition, catheters in urinary tract disrupt normal clearance of microbes from bladder mucosa and voiding. At the tissue implant interface, local immune defenses of host weaken. Therefore, less virulent environmental saprophytic flora can colonize the biomaterial surfaces. Virtually, any bacterial species present in the environment can be introduced into surgical field accidentally and subsequently leads to early acute or late subchronic infections. A study isolated 1027 microbial strains from 699 patients who underwent revision surgery due to orthopedic infections. Just over 3/4 of these strains were attributed to 4 most prevalent species, i.e. Staphylococcus aureus, Staphylococcus epidermis, Pseudomonas aeruginosa and Enterococcus faecalis, while the remaining quarter were attributed to over 50 species. This implies that by targeting these few species can bring about significant impact. Bacteria compete with human cells to reach the surface of biomaterial. If bacteria reach surface faster, they initiate biofilms formation in stages as shown in Figure 4. Figure 4 biofilm development stages for motile rods, e.g. Pseudomonas aeruginosa, and non-motile cocci, for example Staphylococcus aureus, Staphylococcus epidermis. 1. Planktonic single bacterial cells or aggregates of detached biofilm bacteria attached to biotic or abiotic surfaces. 2. Bacteria produce extracellular polymeric substance, EPS, shown as green slime, to enhance adherence. 3. Bacteria expand their own colonies or together with other species. 4. Bacteria maintain mature biofilms in steady state. 5. Single bacterial cells or aggregates with EPS disperse to colonize new surface. Second row of images showed Pseudomonas aeruginosa in these stages under confocal microscope using 4 feet, 6-diamidino2 phenylindole DAPI, chromosomal green fluorescent protein GFP, and live dead back-like kit. Pathogens embed in hydrated protective matrix named EPS or slime to evade host immunity. In addition, biofilm bacteria are more resistant towards circulating antibiotics and environmental stresses like dehydration, UV light exposure or metals toxicity than their planktonic counterpart. Abiotic surface is initially colonized by single bacterial species. As biofilm develops, a menagerie of pathogens, including yeast strains, can be isolated with phenotypic heterogeneity observed for each species. Rapid inter- and intra-species genetic material exchange makes biofilm a reservoir to spread antimicrobial resistance genes. In the light of biofilm crucial roles, preventing its formation hinders pathogen survival. Pseudomonas aeruginosa is the commonest bacterial species isolated for CAUTIs and microbial keratitis related to use of contact lens. Besides, it often inflicts VAP and lower respiratory tract infections in cystic fibrosis and immunocompromised patients. Moreover, Pseudomonas aeruginosa has flagella to facilitate attachment, pili to glide across surfaces, and lipopolysaccharides to stimulate host antigenic response. It secretes alginate, N-3-oxidodecanoyl L-homoserine lactone and exotoxin A for stronger attachment, cell-to-cell communication or quorum sensing and repression of host immune responses. Two main directions in handling dyes are killing pathogens or resisting their attachments. Each method has advantages and disadvantages. Since systemic antibiotics are ineffective in eradicating biofilms, focus has been shifted to local modification of medical devices, aiming to kill the pathogens before they form biofilms. Strategies used can generally be divided into two, i.e., reservoir and non-reservoir types. For reservoir type, the medical devices can themselves be loaded or coated with hydrogels which have been preloaded with antimicrobial substances like antibiotics or bioactive antibodies. Delicate control over the release of these substances in vivo is challenging and may lead to accumulation and toxicity due to poor control. Initially, release rate is very high and tapers off over time due to depleting reservoir. Prolonged exposure to sublethal antibiotics concentration promotes antibiotic resistances. When the reservoir runs out, it needs to be replaced. Non-reservoir type involves three main methods. Firstly, medical devices can be coated with metals like copper. Matthews et al. Showed that metal-bacteria contact disrupted bacterial cell envelope integrity, which subsequently permitted the entry of copper ions so that reactive oxygen species were generated, resulted in cell death. Otherwise, topography of biomaterial surfaces can be modified to confer bactericidal properties. Surfaces could be engineered to resemble those nanopillar arrays found on Clanger cicada, Saltoda claripenis, wings which could pierce Pseudomonas aeruginosa cells upon contact, leading to death. Alternatively, antibiotic molecules can be bound covalently to devices' surfaces directly or via spacer units to aid the mobility of drug molecules. Given the limitations of bactericidal strategies as aforementioned, strategies targeting to resist bacterial attachment, stage 1 of biofilms formation, are developed successively. A recent example was PDMS infused with silicone oil to produce an extremely slippery interface where bacteria could not attach firmly. 0.29 Besides, PDMS surface engineered based on shark skin microtopography had nearly 8-fold less S. aureus coverage than smooth PDMS. Alternatively, monomers containing chemical moieties which resist bacterial attachment can be grafted onto medical devices. Two examples are EDGEA and DMEM, which contain cyclic pendant ring and ethylene glycol, e.g., refer Figure 5. Moreover, DMEM can regulate flexibility of coating. Figure 5, chemical structures for EGDEA and DMEM. Figure 6, mechanism of base-catalyzed PDMS surface modification. Firstly, O, the nucleophile attacks both PDMS and 3-mercaptopropyl trimethoxysilane (MTS), the coupling agent to produce silanoate ions R3CO. This triggers cascades of nucleophilic reactions leading to A, depolymerization of PDMS surface, B, production of MTS oligomers, and C, metathesis, i.e., incorporating various forms of MTS onto PDMS. Sulfur analogues are better than alcohol analogues because they are less basic nucleophiles and generate radicals easily in controlled way. Thin surface modification is preferred to preserve the functional and mechanical properties of bulk material. The reaction conditions like temperature, concentration of reagents used are controlled to reduce undesirable cracking, etching and cross-linking on surface. Methanol, which does not swell PDMS effectively, is a suitable solvent so that thiols are incorporated to the external surface rather than penetrating into the bulk material. The synthesis of copolymers can be achieved via few controlled radical polymerization methods. An example is catalytic chain transfer (CCT), which involves breaking the double bonds of acrylate and metaacrylate groups of EGDEA and DMEM monomers, refer Figure 5, to form random or block copolymer known as poly-EGDEA DMEM, as shown in Figure 7. CCT is chosen mainly to preserve the unsaturated bonds at nearly all termini via mechanism as shown in Figure 8. This is essential for subsequent grafting to thiol-modified PDMS. Moreover, conditions for CCT are milder, using less chain transfer agents and initiator at lower temperatures. This saves cost and reduces the contamination risk on medical devices by those potentially toxic chemicals. Low molecular weight polymers are produced. Figure 7, schematic diagram shows catalytic chain transfer polymerization of EGDEA and DMEM to generate poly-EGDEA DMEM. Figure 8 CCT mechanism. Cobalt-2 serves as catalyst. It abstracts a beta hydrogen atom on a growing polymer chain and becomes oxidized while restoring terminal vinyl groups. The cobalt-3 hydride will transfer the hydrogen to another monomer to produce new growing chain. Simultaneously, cobalt-3 is reduced to its active form again, ready to undergo stage 1 again. There are two approaches for UV-mediated grafting, i.e. graft from and graft to, as shown in Figure 9. For the former approach, monomers form covalent bonds with thiols on PDMS surface, while for the latter approach, copolymer chains form bonds with thiols via radical-based thiol-ENE click chemistry as shown in Figure 10. This is basically breaking a carbon-carbon double bond in monomers and sulfur-hydrogen bond on PDMS surface to form sulfur-carbon bond. Figure 10, thiol-ENE click chemistry begins with I. Photoinitiator decomposes upon UV irradiation to generate free radicals. E. The latter abstracts protons from thiol-functionalized PDMS, leaving reactive PDMS surface to E. react with vinyl termini of monomers or copolymers. IV. The intermediate radicals then abstract protons from neighboring thiol-functionalized PDMS to regenerate reactive surfaces. V. PDMS are coated with poly-EGDEA DMEM grafts. In this study, we aim to compare the ability of PDMS samples with and without poly-EGDEA DMEM coatings to resist bacterial attachment. Simultaneously, we aim to compare graft to and from approaches in terms of resistance to bacterial attachment. End of introduction. Stay tuned to Episode 3 Materials and Methods section. Thank you for watching. If you like my videos, kindly like, share and subscribe to my channel. See you all in the next video.